Good afternoon, everyone. Welcome to the last session of the robotics seminar for this semester. It's a pleasure to have Professor Ritu Raman with us. So Ritu is a DR Beloff Assistant Professor in Mechanical Engineering. She got her bachelor's from Cornell, then was doing a PhD at UIUC, then came here at MIT to work in Bob Langer's lab, and then started her own awesome lab. Ritu's work has received multiple awards, including the NSF Career Award, multiple Young Investigator Awards, and she has also been recognized as Forbes 30 Under 30 and the MIT Tech Review Award. So Ritu, it's a pleasure to have you, and looking forward to hearing more about biological motor control for soft robots. So please take it away.
Great. Thank you so much. I honestly thought there would be like two people here because the work we do is very different, I know, from the usual MIT Robotics Seminar series. So thank you all for coming. I'm excited to talk with you. And since it's still like a relatively smaller group, feel free to like interrupt and ask questions at any time, especially because the kind of work we do is heavily rooted in and inspired by biology. So I'll try to explain the biological context and terms and cues as we come across them. But if you're like, please wait, stop, explain more, interrupt in the middle. That's totally fine and highly encouraged. Okay, so like probably many other talks you've seen, the work in my group is heavily inspired by and motivated by biological motions and biological creatures. So we know, right, both there are beautiful examples in our natural world of the ways in which natural organisms are able to navigate really unpredictable environments. And we know that the reason they're able to do this is because they're leveraging the compliant actuation of skeletal muscle with neural control and real-time closed-loop sensory feedback. And we also know that a lot of synthetic actuators that we currently have are not able to adapt their form and function to changing environments in the way that biological creatures are able to. I emphasize currently because I know that there are a lot of beautiful there's a lot of beautiful research being done both at MIT and outside MIT that you've probably seen of ways in which we are developing new synthetic actuators that can mimic some of this functionality. And the approach that my lab is taking, which is different from sort of traditional robotics, is rather than using synthetic abiotic materials to replicate these functions, thinking about how we can just build biological tissues from scratch using living cells. So that's what I'm going to talk about today. But to kind of orient you on the sort of functional metrics of muscle, living muscle, as compared to other actuators that you might be more used to seeing. We published this review in Science Robotics a few years ago that's like mostly still relevant, though it's a highly evolving field. But if you think about how much force an actuator produces as a function of its size, right, we're used to seeing graphs like this. If you kind of map out where muscle falls as compared to, say, shape memory alloys, pneumatic actuators, hydraulic actuators, things that we're used to using primarily in soft robotics, everything kind of falls on a linear line, right? It's a log-log plot, but still sort of a linear trend where the bigger you get, the more force you're producing, so that's good. The nice thing about muscle is that it's highly scalable, right? So each cell is sort of microscale. You can stack many things to make actuators that are, at the centimeter or meter scale. But you can also operate at the single micron scale if you want and scale largely along that line. The other really nice thing is that you have adaptability, right? So muscle, as you'll see throughout the talk, can adapt to previous cues in its environment. So it can exercise and get stronger. It can heal from damage. And the most important thing, I think, that we often neglect is that these sort of graphs are not accounting for the power to weight ratio that also accounts for the size of the battery, right? So muscle uses a very, very dense and highly biodegradable energy source. It just uses sugar. So if you account for the power to weight ratio that accounts for both the actuator size and the robotic and the battery size, muscle is actually one of the most efficient actuators that we have to date. So these are sort of some of the motivations for using living tissue in robotic platforms. And certainly we're not the first people to think of this idea, right? So one of the seminal examples of robotics that leverages biological tissues came from Kip Parker's group several years ago now, in 2007. So he's at Harvard, and they had developed these sort of soft, thin polymer films on which they were growing a sheet of cardiac muscle, right? So cardiac muscle sort of beats synchronously and autonomously. And over the years, they've sort of developed on this platform where they realized that adapting the form of their robots to the shape of different biological organisms, and then using different types of control schema and agonist-antagonist muscle pairings could be used to make robots that could sort of swim and traverse different environments at the same speed and with the same efficiency as natural organisms like stingray or fish. So these sort of machines leverage cardiac muscle, which is great when you want synchronous, autonomous motion, like swimming. But most of our autonomous locomotion, both in our bodies and in animals more broadly, leverages skeletal muscle. So I'm going to give you a very quick high-level intro into the differences between these two tissue types and why you might want to think about using and building with skeletal muscle instead. So cardiac muscle is for your heart, right? That's the only place it's used, and it's there because you want it to beat all at the same time and you want it to never stop, ideally. If it stops, you stop. But that's not what you want for a robot, right? You want a robot to be able to do something and then like chill out and then do something again later, and then also do things like get stronger over time or get weaker if you're not using it as much. And you also want it to be able to heal from injury. So all of those sort of adaptive behaviors and on-off control behaviors are not possible with cardiac muscle, right? You can't turn it on and off. You can pace it, but that's all you can do. If it gets injured, right, you have a lot of scarring, and that's essentially what a heart attack is. So we know that we don't recover very well from those. Even if you survive past it, you have scar tissue that then impacts your ability to generate force and produce motion. That's why we have things like pacemakers. And then we also, you have some adaptability in your heart over the span of your life, but not much in adult life. But, I think we all have sort of a lived experience of if you went to the gym for 2 hours every day starting today, your body would look very different next year. And if you were doing powerlifting versus training for a marathon, your body would look very different next year, right? So that kind of functional behavior and training is something that's only possible with skeletal muscle. So that's sort of the basic biology of skeletal muscle is that each cell is basically a long fiber. The fiber has these two proteins called actin and myosin that slide against each other. When they slide against each other, the whole tissue or the whole cell sort of contracts in length. And if you stack many of these in parallel, you can make something that looks like a muscle, right? So each cell is an individual actuator, and together they generate large forces. And then upstream of that muscle, you actually need something to trigger that contraction. So in our bodies, that's mostly motor neurons that are sitting there's upper motor neurons that are sitting in your motor cortex or brain. We're not going to talk about those today. They're responsible for motion planning and coordination. We will only talk today about lower motor neurons that sit in your spinal cord. So one motor neuron usually controls several muscle fibers. If it's muscle fibers in your leg, it's usually hundreds or thousands of muscle fibers. So one neuron is controlling many muscle fibers at a time. That construct is called a motor unit, one neuron and the fibers it controls, and many motor units form a muscle. And the size of motor units depends on the type of motor control you need. So in your legs, one motor unit is thousands of muscle fibers. In your fingers, where you need fine motor control, only a few muscle fibers are part of a motor unit, right? So that's a kind of beautiful, nice hierarchical structure that we have, and it's really nice because it's very robust. If one neuron goes down, that doesn't mean that your whole muscle stops working, and a few muscle fibers are damaged doesn't mean that you're impacting every motor unit in place. So that's very nice. We would like to build that. That's what my lab is trying to build. It is very, very hard, right? That's the main takeaway of this talk. So I'll tell you about where we are in trying to build this biological system from scratch and kind of where we're going and how we're leveraging this in robotics. Okay, so that's muscle. Yep.
Multiple motor units kind of overlap in the muscle fibers that they control?
No. So each muscle fiber is only controlled by one upstream motor neuron. However, the muscle fibers that neuron controls are not necessarily physically adjacent to each other in a muscle. So they can be sort of distributed throughout. So this is basically the sort of thing that then means that if I'm lifting this USB versus if I were trying to lift this whole thing, you would be activating different numbers of motor units. Yeah, great question. We'll talk more about also Within each, so there's a higher level thing here that happens where you also have 3 different types of muscle fiber types in your muscle. Some are slow twitch, which means that you generate sort of low forces for long periods. They're very endurance and fatigue resistant. So that's like your postural muscles and your torso. So that means you can sit and stand for long periods of time with minimal effort. And then you have a lot of fast twitch fibers in your limbs, which means that you generate large forces, but you get tuckered out fairly quickly. And then there's some intermediate stuff. It will become important, so you'll see. Okay, so to build muscle in vitro, how do you do this? The first thing you want to do is just think about how you're actually transmitting force from your muscle to some skeleton, right? So most of the time, muscle is tethered to bones via tendons that are stiff. They're less stiff than bone and more stiff than muscle, right? So they're transmitting force through this sort of stiffness gradient interface. And they're always held in a state of passive tension. So this keeps all of the fibers aligned and pointing in the same direction. So when they contract, they're contracting in the same direction. And then the bones will usually have some compliance in the form of a joint, like an articulating joint, like your elbow or your shoulder or your knees. And so that when that shortening happens, you will get some actuation that is larger than what you would anticipate based on the shortening alone. Okay, so I'll talk about tendons and skeletons, but first I will tell you about how we make muscle. So to make the muscle, we use tools there's a broad class of biomedical engineering called tissue engineering, which is like building tissues from living cells. Pretty much everyone in that field uses a similar type of protocol where you take cells that are sort of stem cell-like or progenitors of the tissue you want to make, and you mix them in some liquid goop that has a lot of proteins that are similar to the in vivo environment. So the environment in your body. So we do the same thing. We take mouse cells, muscle cells, when they are what we call in a myoblast-like state. So they're not yet these fibers, but they're single cells. They can then fuse together to form fibers. So they're sort of baby muscle cells or muscle stem cells. Most of the data I'll show you today is with mouse, but we also use human cells in our work, and we can talk about that if you want. All right, so you mix together the cells in your liquid goop. The liquid goop polymerizes into a 3D hydrogel. A lot of hydrogel chemistry here, not super important for you to understand the robotic stuff. Over time, we make these sort of millimeter scale like tissues, right? The cells are the blue circles that you see on the screen there. So this whole thing, 6 millimeters. That screenshot is a few hundred micron squares zooming into the tissue itself. So you have single cells in blue that fuse together to form fibers. The fibers are then stained for a marker of mature muscle, which is shown in green. So you have these long green fibers, and then you see that they're all pointing in the same direction. And the direction they're pointing is also the passive tension that's being imposed on them by the skeleton. This is important, and it turns out that that's highly plastic. You can change the directions that the fibers are pointing based on the mechanical forces you impose on the cells during their development. So we'll talk about that. Yeah.
How long do these cells survive in the medium?
Great question. So one, you have to give them food every day, just like yourself. So if you refresh media for them every day, we typically keep them on the period of several weeks or months. However, we have kept our robots alive for years before. It can be highly variable. So initially, we couldn't figure out why. There was one guy that was just like going, we called him Leonardo, and I fed him every day essentially for 2 years during my PhD, because why not? I was going to lab anyway. Totally fine. And there were many that would just sort of like break or stop working after a period of a few weeks. We couldn't figure out why. We did a study several years ago where we essentially saw that some of the cells are always active, right? They're sensing their environment. Sometimes they get stressed out. When they get stressed out, they secrete these enzymes that hack away at their protein matrix. And when they do that, the tissue breaks apart. So if you put inhibitors of that enzyme in the liquid media, then you can keep the tissues alive for very long, which is what we do now. So now when we get rid of them, it's because the experiment is not done, not because the tissues are not working anymore.
And is there any rebirth of cells? Is it the same cells which...
Yeah, another great question. In our mouse tissues, there is no rebirth. So the way muscles work is like once they get to this fiber state, they're what we call terminally differentiated. They can't reproduce. Usually in our muscles, we also have a population of undifferentiated satellite cells. In our mouse cell line, that doesn't work so well. In our human cell lines, we do have undifferentiated satellite cells, so you might see self-replication there. We haven't yet, but only because we just started working with them. Okay, all right. So just because the muscle looks like muscle doesn't mean that it actually works like muscle, right? So we have to test its ability to generate force and produce motion. Typically, if you want to mimic how or how motor neurons control muscle, you have to open and close a voltage-gated ion channel in the cell's membrane. So that's naturally present in muscle. So if you put two electrodes and put the muscle in between and send an electrical pulse, the muscle would contract. However, when you do that, you can cause a lot of contamination in your media, you cause a lot of bubbles that mess with imaging, but there are a lot of really beautiful optogenetic tools, we call them basically genetic engineering tools, where you can put a light-gated calcium ion channel in the muscle cell membrane, similar to a voltage-gated ion channel. You take it out of algae, you put it into your mouse muscle, and then when you shine light on the muscle, you get the muscle to contract. So here that black band of tissue is contracting. It's around some flexible skeleton that's out of some polymer, and then you can get the muscle to move and generate force. Yeah?
So is this rubber band is getting manually controlled or something, or like they're pumping automatically?
So every time the light flashes, the light-gated ion channel opens, calcium ions flood in, those proteins slide against each other, and the muscle fiber is contracting.
So is it like light? So the light is like activating the muscles?
Yes. Okay, sit with that. We'll come back and we can talk about the biology a little bit more. Okay, so once you can get the muscle to contract, you can get it to do cool things. So the first generation of robots that we made with these sort of ideas were a generation of one-legged, multi-legged robots that could move in one direction, then move in multiple directions and rotate, and then eventually that had like an onboard LED that they were carrying and could be wirelessly controlled and moved towards specific objects. Over time, we and others have used our muscle actuators to make robots that swim and grip and pump as well. You can essentially think of it as any other linear actuator, right? So if you were to turn that contraction into some functional locomotion of some different kind, you could do it in the same way you would with any other kind of actuator. So these are the sorts of machines we were building for several years. Recently, what we've done is said, so one of the kind of disadvantages of working with this type of setup is that the muscle is just like, it's sort of a friction fit. It's just placed around that skeleton. So it can slip and slide. The contact area varies a lot. If it moves down a little bit, the amount that the amount of deformation will change. And also, when you have to put it on a skeleton that's relatively soft in this configuration, right? So usually you make it out of some hydrogel or polymer. That material is viscoelastic, which means that it has a frequency-dependent stiffness, right? And muscle generates its largest forces at highest frequency. So then if you're testing your muscle at high frequency, it's not actually able to deform the skeleton, and you get these highly variable responses. So it was fine for designing sort of our first generation robots just to show proof of concept, but it was not useful for making robust and reproducible machines. So what we've done most recently in this paper that came out last week is look at a way to essentially get exactly the same performance every time from muscle and to put it on a rigid linear elastic skeleton instead that more sort of naturally mimics its native environment. So the way we did this is by first modeling what happens when you just take muscle off of a skeleton and just have it floating in liquid. It won't last like this for long because, again, the fibers do need some mechanical tension to be kept in alignment. But if you take it off the skeleton, immediately take a video of it, you can sort of see the sort of maximal strain that it would generate. And then you can sort of decide, this is a strain limited actuator, so you can decide what is the stiffness of spring that I can couple this muscle to sort of maximize the motion. So we've been designing flexures, which are basically compliant mechanisms where they are linear elastic and you can program their degrees of freedom of motion, right? So we have essentially a 3D spring. We put our muscle on it and we design it such that we can carefully tune the ratio of the flexure stiffness to the muscle stiffness. And this is important because essentially, you can look at the paper if you're interested in the modeling, but essentially if your flexure stiffness is much less than your muscle stiffness, when the muscle contracts, it will move the most, right? All the flexure is doing is sort of providing it a guidance on like what direction it can move, like X direction along the muscle fibers itself, but it's providing no resistance to contraction beyond that. As you move farther and farther up and your flexure stiffness gets very high, you can extract large forces out of your muscle because it's really pulling against it and transmitting force to the skeleton, but you'll get a smaller range of motion. And then somewhere in the middle, you can sort of maximize your work output. So you can vary your ratio of flexure stiffness to muscle stiffness to get a variety of different outputs. And you can also increase the sort of accuracy and precision of your movement. So I mentioned that if you have muscle on these sort of flexible beams, they're very, very sensitive to exactly how and where you place the muscle on the skeleton. So you get very small movements, right, on the order of like tens of microns that differ a lot between devices. If you take those exact same muscle tissues from this experiment, take them off the beam and put them onto our flexures, you can improve the movement by about 5 to 6 times. So it increases by over 5x. And you get the same force output every time because the flexure itself is compliant, but the post that the muscle is on is rigid, right? So it's insensitive to exactly how or where the muscle is placed on it, and it can transmit the same force every time. So now we get these really accurate and reproducible and maximized strain outputs from our muscle actuators, and we can use this to understand more about the dynamics of muscle contraction and how you might sort of tune it over time. So in a single muscle contraction, usually say you flashlight on it once, you would see some time for the muscle to respond, that's very short, some time to get to its peak force, some time that it spends at peak force, and then a relaxation curve. For many generally when people make muscle in petri dish environments, they're not interested in anything beyond this because they're sort of mimicking the natural biological environment where a neuron fires, usually for like milliseconds, and then the muscle contracts and has this response. We were like, okay, well, there's no biological limitation in this case. So what happens if we just keep the light on for longer, right? Can the muscle hold at peak force? And it turns out that it can. And honestly, I'm surprised that people hadn't tried this before, but I think there was just like not that many people using muscle for robots, right? So they're not asking these kinds of questions. But essentially what we see is if you change the duty cycle of your signal, you just keep the light on for longer, the muscle does in fact hold at its peak force for a longer period of time, and then it relaxes back down. There's also a frequency-dependent response in muscle, which I should have mentioned. So usually because of this characteristic response time, right, that relaxation time is something you don't, and the contraction time, they're not things you have control over. Like those are natural properties of muscle. So if you have a low frequency signal, the muscle contracts and it has time to relax before your next input comes in. As you go at higher and higher frequency, your muscle doesn't have time to fully relax before it contracts again, right? So you have a reduced dynamic range of motion, and at very high frequency, you basically just hold at peak force, and this is called tetanic contraction. So in natural muscle, that usually happens at about 20 hertz or 40 hertz. You have to get up really high, but we kind of showed that even at like fairly low frequency, like 4 hertz, for example, if you just keep it at its peak force for a longer period of time by changing the duty cycle of your input, you can induce tetanic contractions at very low frequencies and essentially decouple the magnitude of motion that you can get from the time you hold at peak force. So we now have the ability, in the past when we were like, okay, I want my muscle tissue to hold at a certain frequency for a very long time or hold at a certain force for a very long time, that then meant that it couldn't move very much. But here, even at low frequency where it moves a little bit more, you can sort of keep it at its peak force for a longer time. So decoupling those variables is important because, again, we're trying to think about how do we design mechanical systems, controllable mechanical systems that are powered by muscle, and now we have the ability to do that. Okay, and then is this important? Yeah, I mean, okay, this is not super important, but one thing we've sort of looked at also is characterizing sort of force, work, and power outputs at different frequencies. And what we've seen is that because the relaxation speed of muscle is like a passive biological process that we don't have control over, at higher frequencies, you have sort of a higher variability in your work and power output because you don't always know exactly where you'll be in your relaxation cycle before the muscle gets its next input to contract. But on average, at higher frequencies, you also do generate more power. So for example, when we've made our walking robots and we stimulate them at higher frequencies, they walk faster. They're maybe more variable in speed, but they walk faster. So there's always a trade-off there. Yeah.
Do you see these attributes changing with different sizes of actuators that you make? And do you think that if you scaled it up to, like, say, a human-sized arm or whatever, you'd you expect it to act similarly, like obviously corrected for mass and sizes?
Yeah, I would expect them to act similarly, largely because as long as, like, the individual actuator is each cell, and all you're doing is changing the number of them there's no other like inherent process that would be taking place that would, I think, change their response at that scale.
Even though you might be controlling them using a different signal than like the brain would use, for instance.
Well, oh, that's a good question. So one thing is that light doesn't penetrate very deep into tissue. We have seen that when we make much thicker tissues, then when you stimulate them with light, it doesn't activate all of the fibers in there. And so in that case, you see some difference. But we are actually moving towards neuronal innervation of our actuators, which you'll see at the end of the talk, and then that will sort of be mediated there. Great. Okay. So, and then the last thing that we were able to sort of observe with this initial flexure study is looking at fatigue and endurance response of our muscle actuators. So we essentially looked at how they respond to continuous stimulation for 30 minutes at different frequencies. At 1 hertz, what we saw is essentially you get some variability in the force velocity curve that they're traversing over time, but they don't really get tuckered out, at least at 30 minutes. But at 4 hertz, you see a significant drop-off take place over 30 minutes of time. And now, what are the mechanisms of fatigue here? Well, you could just say, well, it's moving 4 times as many times, right? So what does that mean? It could be that there's in the body, when you get tired, there's multiple reasons. One thing is like a lack of nutrient availability, right? So there's glucose, there has to be glucose that is turned into ATP that can power muscle contraction. If there's no glucose, then you see fatigue. However, this liquid tissue, it's a 6 millimeter piece of muscle. It's sitting in a 50 milliliter vat of liquid with a ton of sugar in it. So we don't think that that's the case here. The other reason you have fatigue in your body is because your neurons get tired of firing. In this case, there's just a optical fiber, right? So that's not true here. So then that sort of leads us to believe that the mechanism of fatigue here is due to the mixture of slow twitch and fast twitch muscle fibers in our tissue, which I mentioned in response to a question earlier, where these were our muscle tissues we exercise them regularly, so they're trained at low frequency, so what we call like 1 hertz every day for about 30 minutes, which means that they probably have a lot of fibers that are responsive to low frequency stimulation and are used to working at that stimulation. We have some early evidence now in our lab to suggest that if you exercise train muscle at higher frequency, then they're more fatigue resistant at high frequency because you can mediate the fiber type distribution in there. But very early days, so we're digging into the mechanisms there. Okay, and then the last part about mechanism design, and then I'll show you some more biology work, but please don't run away when I do. The last part we want to do is, like, right now we have a good way of making muscle and we have a good way of making flexible skeletons, right? But the way we're doing things still is like taking a rubber band and like stretching it around bones, which is not how our bodies are designed. Our bodies are designed to have these really beautiful bone, tendon, muscle, tendon, bone interfaces with this stiffness difference in between. And that way you're not wasting all of this. If you do this, you're wasting all of this muscle tissue around the edges that's not really doing anything for your system and is also not particularly well aligned. So we were, wondering, how do we make tendons? One way is to make them out of cells. The thing with biology is anytime you move to a new cell type, you have to have expertise in that cell type, right? And my expertise is in neuromuscular systems, not in tendons. And I figured, all right, that would be a harder way to try to do this. Another way to do it is to just think about developing some sort of tough hydrogel that could serve as a interface between the muscle and the skeleton. And I have a lot of expertise in hydrogel chemistry, so I was like, I'll do that instead. So we worked with Xuanhe Zhao's lab here at MIT in our department in Meki. And they have a range of tough hydrogel systems where they essentially put a chemistry on there that sticks really well to most proteins, which means that if you take this hydrogel adhesive and stick it, to basically place it on top of our muscle for a few seconds, it forms a really, really tight bond that is resistant to forces that are orders of magnitude higher than the forces that are being generated by muscle. So we can stick it to either end of our muscle and then put it on top of our skeletons. And then when it's kind of hard to see because the hydrogels are transparent, but this part is a hydrogel there. This part is a hydrogel here. This is a muscle tissue that we made it like a normal ring and then just basically cut it and stuck it on either end. And then this is the rigid skeleton that it's attached to. And it took us a really, really long time to figure out how to do this, both how to get the chemistry right, but also what is the ideal stiffness of tendon that transmits force from the muscle to the skeleton, and also what is the ideal pretension that you have to put in the system such that there is not too much slack in the muscle, but also it's not being torn apart. So we have that data. I can't show it to you because if a grad student is doing a lot of experiments in the lab, they can't also have beautifully made graphs, right? These things happen in phases. So just believe me, it's there. But the outcome is that we can now have these muscle, tendon, bone structures. And now we're thinking about how we leverage this sort of design space to make gripping, walking robots that do fun things. All right, okay, so that's robotics. So I said the next thing that we want to do in our robots is to integrate neural control, right? And we tried to do this for many years and ran into some difficulties, but what we have, I have a sense of the time, I just wanna, 3:38, okay. 3:38, is that the time? Yep. Okay, great, okay, I'll talk fast. All right. So we didn't know how to do it for a long time, but then my lab also does a lot of work in medicine and understanding how to preserve and restore mobility to humans who are suffering from neuromuscular disease. And work that we did in the medical space ended up informing how we might be able to integrate neural control into our robots. So high level, what we did to do this is we basically take a mouse, we're very mean to this mouse, we take a huge piece of its leg muscle out and we implant our muscle actuators at the site of injury. And the idea of doing this is that over time, that muscle will integrate with the surrounding animal's muscle as well as its blood vessels and its nerves, and then the mouse would be able to use it and walk around. So the first thing we do is we just compare how our muscle looks with the native mouse's muscle. So I want you to just focus on these green panels right here. These circles that you see are muscle fibers in cross section. So you have a long fiber here and we're cutting like this. This is the left leg of the mouse, so that's healthy, never been touched, just a native muscle. And then that's the muscle that we've made and implanted. So what you can see is that they look fairly similar, right? You have these densely packed circular fibers. And then ours also have these pink fluorescent tags. And all that means is that those are our light responsive cells. So that means that our genetically engineered cells are in fact at the site of injury, not the native muscle cells. The other reason we know this is that then when we shine light on the graft through the skin of the mouse, we can control the graft actuation and the sort of like kick response of the muscle. So this is not the mouse trying to kick, right, but us sort of forcing that muscle to contract and then transmit that force to the mouse's skeleton. And so when we compared the electrical activity of our grafts to the native mouse's muscle, we can do this via things like electromyography. What we saw is that essentially they're sort of equivalently electrically active, which means both muscles are equally healthy. But just because you have equally healthy muscle does not actually mean that you can use it, right? Because if there's no innervation, no neural control of the muscle, then you can't use, the mouse can't do anything about it. Only I would be able to control it with the LED. So to assess whether there's nerve control, yeah.
Because if you at least with organ transplants, right, the body can attack it, thinking of it as a outside particle. So I'm wondering if you run into these kinds of issues, or how do you make the body believe?
Yes, that definitely can happen. So in this particular case, we use immunocompromised mice because the focus of the study was more looking at exercise. If I were to think about using this as a transplant for a person, we would use, so we have induced pluripotent stem cell derived grafts that we make. So this means that we would take skin cells from you, expand them into turn those into stem cells, differentiate them into muscle cells, make a muscle graft based on those, and then put your cells back into you. But then there would be some immunogenicity still, probably based on other materials. But yeah, so that's definitely something that one would have to think about long term. Okay, so we wanted to look at whether the mouse actually has control of the muscle grafts as well. So we essentially measured how long does it take a mouse to run from like one end of a room to another end of a room. And we saw that those that had exercise grafts were essentially running at pre-injury speeds within about 2 weeks. And in the scale of this sort of injury, this is a sort of thing that mice would never be able to recover from on their own, and with other therapies would maybe recover around 50 to 60% over several months. So a 2-week recovery, full recovery period is like very, very good. And we wanted to figure out, well, The only reason that would be true is if there were actually nerves that had grown into the muscle, right? So we wanted to figure out how that would be. I'm not going to go into the biological tools and assays we use to do this, but high level, we use something called phosphoproteomics. You take your tissue, you freeze it, you crush it, you run it through a mass spec, and it looks at what proteins are being turned on or turned off. That's all you need to know at this level. And what we learned is that when you exercise muscle tissues, you appear to upregulate the activity of proteins that play roles in growing new blood vessels and growing new nerves, as well as like 1,000 other things. And this effect size is enhanced by the amount you exercise. So the more you exercise, the more you see this innervation happening. So this is, nice and convincing, but in an animal, it's like very, very difficult to prove to anyone that truly muscle contraction is driving nervous control, because there's fat, there's tendons, there's bones, there's immune system, there's a million other things that could be causing this crosstalk. So we wanted to then take that system outside of the lab and essentially think about how we might be able to look at muscle-nerve crosstalk in an in vitro or petri dish context. Actually, maybe I'll scroll back. One more thing I'll talk about now. Okay, so we learned that thing in animals, and then we decided, okay, we want to put neural control into our robots, right? So in 2017, right at the end of my PhD, I was like, okay, I know how to make muscle, so on top of the muscle, I'm gonna stack a bunch of motor neurons, and then I'm gonna let the motor neurons grow towards the muscle, form functional connections, and then they will twitch. And when we tried that, I don't even know if you can see it, but what I'm trying to show in that video is that you get this sort of very tiny twitch response, right? So when you stimulate the neurons with a chemical that makes them fire, you get a very, very tiny twitch in your muscle. But if you compare that to the amount of twitch that you want to see, it's like 1%. So what this tells us is that basically if you have a muscle tissue, imagine my arm is a muscle tissue, right? Or it literally is muscle. So nerve is sitting on top of here, and it grows towards the muscle, and it innervates like 1 or 2 cells, and then it just sort of quits. And it doesn't burrow further into the tissue. So when I stimulate the neuron, I might get like a little bit of twitch here, but I'm not getting twitch of the whole muscle. And we could not figure out how to get the nerves to grow farther into the tissue for a long time. But then our animal study told us that when you exercise muscle, you might be able to enhance innervation or growth of nerves into the tissue. So now we wanted to see if we'd be able to do this in our platform. So what we've done is we basically grow a 2D layer of muscle on a soft gel, and then we have a 3D spheroid that's sitting on top of that gel. So that spheroid has all motor neurons on it. So then we exercise the muscle, we use a lot of computational tools to track the muscle deformation over time, see it getting stronger and exercising, and then we look at how the nerves grow out. The first thing we do is we take, when the muscles are contracting, they're secreting a lot of factors into that liquid media that they're sitting in, and those factors, people believe, might encourage nerve growth, but nobody had actually definitively shown that. So what we've done is shown that if you compare the top row of neurons growing normally versus the bottom row of neurons growing when they're supplemented with these secreted factors from exercising muscle, the neurons grow a lot more, right? So all those, the way neurons look is they have a cell body, and then they have a long axon that projects outward. So you can see all these long green lines, there's more of them, they're growing faster, and they're growing farther away from the spheroid in response to these secreted chemicals from muscles. I won't go into, again, the biology here, but we use a tool called RNA sequencing to look at how all of the RNA inside the cells are being impacted by exercise stimulation. And what we see is in that neurons that are stimulated with exercised muscle look very, very different from regular neurons. And the way they look different is that they have upregulated signaling related to axon projection and guidance, neuromuscular synapse formation or connection formation, and signaling between neurons. So they're more mature and they grow faster, which just sort of corroborates what we see when we take pictures of them. And then the last part of this study is looking at the mechanical impacts of exercise. So many cells, most cells in our body are responsive to chemical and electrical cues in their environment, but they're also responsive to mechanical cues in their environment. And exercise is a very mechanical process, right? It involves a lot of motion that then causes stretch of tissues that are embedded inside muscle. So what we did then is then in the substrates that we're growing our tissues on, we put these magnetic microparticles that we could actuate with an external magnet. The first thing we did was just show that if you grow muscles on these actuating substrates, you can actually program the direction of the muscle fiber alignment. So I mentioned earlier that fiber alignment is very responsive to passive and dynamic tension cues. So this is one of the learnings from that. Do I want to tell you more about this? Well, the high level here is that when you have highly aligned muscle, you get these really beautiful defined contractions. And when you have unaligned muscle where some actuators are pointing in this direction and some actuators are pointing in that direction, you get these very asynchronous beating responses. So you can use this to program anisotropy into your muscle tissues and get beautiful coordinated responses. But also turns out that if you look at the mechanical impact of exercise on neurons, even completely in the absence of muscle, you can make neurons grow much faster and farther just by stretching them as they're growing. So if you dynamically stretch the neurons, you encourage them to grow more, and then you can get them to potentially grow deeper into your muscle tissue, right? So now we're leveraging these sort of learnings to grow neurons and muscles together. I don't know if you can really see that image, but the red stuff is the muscle, the green stuff is the neurons. They grow towards the muscle, they form connections, and then when we fire the motor neuron, we can see which fibers it's controlling and which ones are contracting. So we can map the motor units that are forming and control the number of motor units that are forming through directed exercise cues. All right, so that's kind of a taste of the sort of things we do in my group. That I mentioned some of the stuff related to robotics. Other things we're doing in the robotics space are integrating, thinking about, well, making different kinds of robots, not just walking, but gripping and swimming, but also thinking about how we can integrate real-time feedback control into the systems. When we are making lots of robots, we're also thinking about manufacturing and fabrication. So we're doing a lot of 3D and 4D printing of biological actuators to make sure we can scale them and make them more reproducible in form and function. And then you saw a little bit of this, but a lot of the work in the lab is also focused on making human cell-derived tissues and using them to model injury and disease and develop new therapies that restore mobility. So with that, I'll just highlight the group and the grants, and happy to answer any questions that we didn't get to.
But did you see a difference in the neuron growth between the actually exercised neurons and the magnetically actuated neurons?
Yeah, that's a great question. So of the protocols that we tested, so we tried two different kinds of biochemical stimulation, what we call it low intensity and high intensity exercise. There was definitely a dose dependent increase, so more exercise, there was more biochemically stimulated neuron growth. Mechanically stimulated actually matched we tried like 1 hertz for 30 minutes. That actually matched the effect size of high intensity exercise from biochemical stimulation. If we tried different frequencies, different durations, I'm sure that can be modulated even further. But I think the high-level takeaway is that they are likely equally important and likely synergistically important. And the problem is that most of the time people only think of secreted biochemical cues. So adding this additional component, I think, tells us that they're probably combining both of them can enhance the effect that you see even more.
Questions? Can you use the mic?
Thanks for the talk, Ritu. My question was, I guess, kind of related to a comment you had made earlier in the talk about scaling up the system. More specifically, I'm thinking about, as I understand in biology, these tissues would be developing kind of, you'd have like co-differentiation during embryogenesis of the neurons, the tendons, the muscles, the skeleton, like all together into one structure. And then growth over time. So there's like a whole like time-based component of self-assembly. And these approaches are kind of like differentiating one piece and then adding another piece on top, kind of like stacking elements. And then I assume later assembling together for bigger structures. And I wonder how those two approaches would differ, both in terms of like feasibility for this current approach versus the possibility for larger structures and when this approach gets limited.
No, that's a great, like, sort of grand philosophical question in our field is, like, does it make sense to go bottom up or top down, right? So I would say the sort of things that we are working on are mostly top down, where we make the components and then attach them together. It's kind of probably because I trained as a mechanical engineer, so that's how I build, right? If you see, there are a lot of people in developmental biology that are kind of emerging in this space, and they do sort of everything bottom up, where you start with a clump of stem cells and you differentiate different. My friend Sam Kriegman, I was just on a Zoom call with, he's pioneered this type of robot where you start with stem cells and then some of them turn into connective tissue and some of them turn into cardiac muscle, and depending on how much is cardiac muscle and where it is, you can sort of make the ball move in one direction or another direction. That's also a valid way to make systems, right? I would argue that right now that's a little bit harder to scale to the sizes that we need to be able to make, because we haven't quite figured out how to go from like embryo to large scale structure. The other problem, or not problem, but I think concern with that is that twofold. One, I think we, and probably many of you, we have access to many like non-biological materials that do very cool things, right? So ideally, I think that when we're making biohybrid robots, we should be using biological components when they make sense and synthetic components when they make sense. So this approach allows you to do that. It also then potentially avoids any possible ethical concerns of if you made something that was truly all biological and self-replicating, then you eventually get to something that could be closer to an organism of some kind. And then you have to think about controlling its release into the environment, I think, much more cautiously. That's like a 50-year thought process, right? But I think that would be a concern there.
Ritu, this was a fascinating talk. I'm curious, there's other set of people who are trying to build artificial muscles, but using DEAs. So I'm wondering, how do you compare and contrast, these two approaches going forward, and what, in your opinion, are pros and cons?
Yeah. I mean, I think, so again, I'll emphasize, like, because I'm a mechanical engineer, right, I think that there's a bunch of synthetic materials that are really amazing and we should keep using, right? If you talk about, it's a small field, but if you talk to people who trained more in biology, they're like, well, biology is the best and it solved all the problems and there's nothing you can do. But I think we've developed things where we are better than biology in some systems and we should use them when it makes sense, particularly when we want to make things that are maybe cheaper. That would be a great time. But I think there are some environments, right, where you might be designing for an environment where you don't actually know what a robot is going to face. And so you really want to have that real-time adaptation and ability to get stronger or weaker or change your performance needs as needed. So all of our DoD grants, for example, are funding like exploratory robots. So if I wanted to really put something into an unknown environment where I might not know all the cues it might face, then a biological system might make a little bit more sense. Another situation is obviously also in medical applications where you want things that are very soft and warm and humid and highly energy efficient and compatible with surrounding tissues. So if you wanted to think about deploying robots, either surgical robots or implantable robots, making them out of biological materials might be a little bit of a safer bet. But I think not always and sometimes.
Any more questions from the audience? Maybe one last one. So what will it take to scale up from the scale that you have now to maybe having a mouse level organism which has been built, all from artificial muscle?
Yeah. So one thing is there's two ways to scale up, I would say, the muscle size itself. One, we literally could just make bigger things. We can make them right now. However, if you don't have blood vessels in there, you can't keep all of the cells inside your tissue happy. So I'm trying to get a grad student to join this fall. I think that'll happen, where we're developing sort of vascularized muscle constructs that we can make a little bit bigger. But then also I think you need to have active pumping of the vasculature, you need innervation, and then if you're making an organism scale thing, there are also a big component that I've missed, right, is any kind of feedback control. So sensory neurons are what usually detect the rate and degree of stretch in our muscle, and sensory neuron culture is very, very difficult right now. So until biologists solve that problem, I think thinking about how we can have flexible strain gauges implanted in our muscle and then using output from those to control neurons would be important for making a organism scale structure that actually does something useful.
So let's thank you for the wonderful talk.
Thank you.